All right, so good afternoon, everyone. My name is Cibu Thomas. I'm a staff scientist in the section on learning and plasticity in Dr. Leslie Angolida's section. And so I'm going to be talking about imaging changes in brain anatomy. This is the first time I'm giving this lecture, so I thought of starting off with what I think is the scope of this lecture. So everything that I'm saying today is mostly a cognitive neuroscientist perspective on imaging brain changes. So I think the material that I have put together is probably going to be useful for people who are starting or in the middle of a project looking at structural brain changes. That said, I think many of the ideas are extendable even to looking at functional brain changes. And I'm assuming that, since this is the tail end of the entire series of seminars, you're familiar with the MRI images and the jargon to some extent. But if you have any questions, please feel free to stop me. So first off, to start off, let me ask you if you think there's something common that ties all these emojis together, some common feature about these emojis. Any guesses? Here's a clue. Come on. No? So these are all factors that change the brain, right? So growing up, growing old, just about, if you look at everything, just about everything changes the brain. In fact, just sitting there, your brain's changing even as I speak for many reasons. And what we are trying to understand is, like, can we pick up these changes with MRI? And what can this tell about brain and the behavior and so on and so forth. And so there are a whole host of questions that you can ask. So the reason why I think imaging brain changes is important is because if you understand how and what changes the brain, what are the factors that change the brain, then we can, for instance, slow the effects of aging or some degenerative disease like Alzheimer's, Parkinson's, and so on. If there is a disease and if you have a treatment technique, you want to be able to measure the efficacy of that treatment strategy. So you need to be able to image the change in the brain, for instance. At a larger scale, this can also help guide public policy on promoting good mental health and so on. So to study the living human brain, you can break it down into two dimensions. One is the temporal dimension, because things are happening in the scale of from probably years in the case of development, or months, weeks, depending on if it's learning or training and learning how to juggle and so on and so forth. Or it could even be at the level of milliseconds, where you have things like repetition priming and so on. So that's the temporal dimension. There's also the spatial dimension, the scale of the changes. So you can have like changes at the whole brain level, which would be the case with a developing infant's brain. Or it could be at the level of certain cortical or subcortical structures. It could be at the scale of millimeters or microns. So what's really powerful about the MRI is that it allows you to map these structural changes in the scale of millimeters to whole brain. You have a really nice span. It is safe, it's non-invasive, it's also translational, meaning that anything that you develop here at NIH, a novel pulse sequence that helps you look at a certain facet of the brain, can then be taken into to the clinic in a hospital, for instance, and you can try it out. So it's translational. But what I think is one of the most powerful properties of MRI is that it's multimodal. What I mean by that is that with the same device, you can program a sequence, which in this case, a T1 weighted image, which gives you very nice gray matter, white matter, and CSF contrast, and you can compute volume and other measures like cortical thickness, gray matter density, and so on. You tweak that sequence and make it sensitive to deoxygenated hemoglobin. You now have task-driven fMRI or resting state fMRI. You can look at how spatially distant regions are communicating to each other. If you tweak it and make it now sensitive to the diffusion of water molecules, then you have a really novel way of looking at tissue microstructure, looking at the major fiber pathways in the living human brain. So this is what makes MRI so powerful. So this is the structural aspect of it. And in terms of the temporal dimension, you can then look at behavior at the scale of milliseconds or years. And now that you have a measure of behavior and a measure of structure, you can now run correlations and look at causality and so on and so forth. So this is what, and so this is how one can image brain changes and understand what it means in terms of behavior. So the plan that I have for today is so review some of the popular MRI methods that are used for measuring brain changes. There are a whole host of them, from T1 weighted images to magnetization transfer, but I'm going to focus entirely on T1 weighted images and diffusion weighted images. And then I'll talk about what we know about the limits. And one of my mentors has always told me this: the devil is in the details. And so it is important to know the details. So we'll talk about that at the level of acquisition, data processing, inference, and how we can overcome some of these limits. And finally, what I think is one of the most important tools for imaging brain changes. I'll keep that a suspense. There's another question coming up. All right, so first off, some popular MRI methods. So here's a T1 weighted image. Takes less than 6 minutes. As I said, it gives you a really nice white matter contrast, gray matter, and CSF contrast. What makes it powerful from a radiologist perspective is that in less than 6 minutes, you can get a very nice picture of the whole entire brain, and you can pick up subtle lesions, like in this case, a neuroinflammatory lesion. But you can use the same image, and you can automatically derive anatomically meaningful measures of volume. So from a standard T1 weighted image, you can strip the skull, segment it into different tissue classes, parcellate it, and then get volume measures of different cortical and subcortical structures. You can overlay it on a surface and then compute subject-specific measures of the volume of various cortical areas. You can take the same image, process it through a different pipeline, and you would get measures of cortical thickness, where here you're drawing the line between the gray pia boundary right over here, the gray pia boundary, and the pia white matter, the gray white matter boundary, and this distance is the cortical thickness, and you can compute that measure. There are also other metrics like surface area, gyral height or sulcal depth, curvature measures, shape measures, and so on and so forth, which can all be useful in terms of studying different patient populations and so on. That same data can be processed using other packages like voxel, in a voxel-based morphometry style, and you can compute measures of gray matter density or gray matter volume, you can do voxel-based statistics, and so on and so forth. So these sort of, at the sequence level and software level, there are so many developments which have made T1 weighted morphometry a very powerful tool for brain research. So in the literature, there's a lot of studies that have looked at things like, how does cortical thickness change as a function of age, where does it change, and so on and so forth. How does cortical thickness change in autism relative to controls, and does things like memory training, does it change cortical thickness in elderly individuals, and so on and so forth. So that's T1 weighted morphometry. Let's take a look at diffusion MRI. So what I'm showing you here is a T2 weighted image acquired from a patient who started presenting with symptoms of stroke, and this was taken 3 hours post onset of the stroke. What the radiologist found with the T2 weighted image was a chronic infarct in the subcortical white matter, which was chronic so it's been around for a while. But when they acquire the diffusion image, now this is not as spectacular in terms of spatial resolution as T2, but if you look at here, what the radiologist could pick up is that there is a decrease in the water diffusion, which suggests that there is an acute infarct extending from the temporal lobe to the frontal lobe. So one of the most widely used method in stroke is diffusion imaging. So because it tells the clinician what's the sort of rate at which the stroke is sort of impacting the brain and behavior and so on and so forth. Now that's the clinical world. From a research perspective, what makes diffusion so powerful is if you consider this T2 weighted image, you can see that the white matter is basically flat. right? I mean, you can see white matter is dark, gray matter is sort of gray, and the CSF is bright. What diffusion does is this, right? It's that same image. Now you have so much of information about the underlying white matter. It tells you that there are fibers here that are going right-left in the right-left direction. These are going anterior-posterior. The blue means it's going inferior-superior, and so on. And so that blob of white matter that you saw here can now be segmented into U-shaped tracts and big, large fasciculi that are projecting in the anterior-posterior direction. So it's a very, very powerful technique that has completely changed how we think about the brain. So what's going on in the guts of diffusion imaging? So the fundamental idea is basically we're looking at the random walk of water molecules. So if you take a beaker of water, drop some ink, it's going to diffuse in an isotropic fashion. We call that isotropic diffusion or homogeneous direction. But if, on the other hand, you have restrictions around it or hindrances, the walls of that cylinder is now going to prevent the diffusion from moving in certain directions. So in the case of white matter or large bundles of white matter, the diffusion is highly hindered. And so what you have here is, if you think of these as little water molecules, they're repelled over here, and therefore they tend to move in this direction, the direction of the, let's say, the axon, right? And from this, you can compute something called as a diffusion displacement profile, which mathematically, it looks like a three-dimensional ellipsoid. And what you get is three diffusion coefficients, which describe how the magnitude and the direction of the diffusion. And the beauty is that with this three diffusion coefficients, you can compute a whole different set of maps. So here's a fractional anisotropy map, which is a sort of a summary description of how coherent the white matter is. Mean diffusivity or trace can be thought of as a proxy for how dense a tissue is, for instance. The directionally encoded color map gives you a sense of what the different fiber architecture is. Is it going anterior, posterior, and so on and so forth. Axial diffusivity describes the diffusion that is going in the principal direction. And radial diffusivity is the diffusion that is in the direction orthogonal to the principal direction of diffusion. And so these have been suggested as proxies for measuring things like myelin and so on and so forth. So this is what people have been using if you're living in the world of diffusion tensor imaging. But more recently, there are other novel methods of describing diffusion. With diffusion tensor, you have the limitation that you're describing diffusion, assuming that the water molecules tend to diffuse in a Gaussian distribution. But that is not the case in brain tissue. And so people have come up with ways to describe, to make a more complex, a comprehensive categorization of the diffusion displacement profile. And one such attempt was this thing called the MAP MRI, which stands for mean apparent propagator, which looks something like this. So if you have, so in the case of a diffusion tensor, if you have a situation like this where there are two fibers that are crossing orthogonally, you would get a pancake-shaped diffusion displacement profile because it doesn't have the ability to resolve those structures. Whereas with MAP MRI, you can resolve this and probably much more sophisticated, so complex fiber architecture. But in addition to resolving the local white matter anatomy, MAP MRI also produces a host of really interesting diffusivity measures. So for instance, these are all the DTI parameters that I mentioned, mean diffusivity, fractional anisotropy, and directionally encoded color map. But you can also look at the degree to which the water diffusion is non-Gaussian, the non-Gaussian in the parallel direction, non-Gaussian in the perpendicular direction. We don't know exactly what this, how this relates to the local anatomy yet, and people are doing a lot of work trying to understand what this means in terms of cellular anatomy and so on. But one interesting thing is the return to origin. So RTOP means return to origin probability. So that describes so if you have a water molecule that is in a particular spot, and if the return to origin probability is very high, you're going to have very much more brighter pixels here. So where would that be? And that would be in a place where the water diffusion is highly hindered. And that would be, for instance, in in the corpus callosum, in the splenium of the corpus callosum, where it's highly myelinated and so on. So it gives you a very interesting understanding of what the local architecture is. Now, beyond this, there are other methods like diffusion kurtosis imaging, and there's also another a method called NODI, which stands for Neurite Orientation, Dispersion, and Density Imaging. Now, all of these are at the heart of it, they're all diffusion imaging, but there are you need to make some changes in the sequence and so on to be able to actually acquire some of these to generate some of these maps. These are, of course, this is a talk on its own, but if you want to learn more about these novel measures, I would highly recommend that you take a look at this review by Elizabeth Hutchinson, very recent paper, which covers all of these things and how it's useful for looking at TBI and so on and so forth. So please have a look at that. So the other cool thing about diffusion is, as I mentioned, it allows you to look at the large white matter pathways. So here's a single slice of a diffusion-weighted image. It's axial image. This is where the eyes would be. This is the back of the head. What you have here is the ventricles, and this here is the splenium of the corpus callosum. I'm going to blow up this. And since this is CSF, the diffusion is again likely to be isotropic. So you're going to have homogeneous diffusion, whereas in these regions, the diffusion is likely to be highly anisotropic. Now, here's an expanded view of it. So the other thing that you see is that there's a coherence to the local architecture here. And this gives you the ability to then project streamlines, which in turn gives you an ability to sort of extract white matter pathways in the living human brain. And then you can compute different measures like the tract volume, fractional anisotropy of a tract, mean diffusivity, radial diffusivity of a tract, and so on. So with this, as you can see, so diffusion MRI is also another powerful tool for brain research. People have been using to look at it to study how, as a function of brain development, what is changing, what are the pathways that change, what are the properties that change, and so on. Longitudinal changes in the corpus callosum following pediatric TBI, and also with brain training. With 2 hours of training, some have suggested that there are changes in the parahippocampal cortex, which correlates with the now newly acquired information about the spatial map of a race of a video game race layout. So that's the outline. So that's just a brief and a quick overview of what these methods are and how we can, how from that principal idea, studies have been generated, some new knowledge has been created, right? So now let's look at what the limitations and what you need to know in terms of understanding the details. And I'm going to break this into scanner-specific issues, MRI sequence-specific issues, subject-specific issues, and so on. And I'll also walk you through how I personally have been able to overcome some of these limits. Right. So now this is something that we are all familiar with, a ruler. And if I have, with this ruler, I can measure the thickness of my index finger, and it doesn't matter whether I take this ruler to Russia, to India, it is the same method. It's not going to change. You're going to get a very precise measurement of something that you're measuring in a linear distance, right? Unfortunately, the scanner is not something like that. It's very Goldilocks. It's a very sensitive piece of machinery. Obviously, you're measuring the brain, so it requires a high amount of complexity. But what studies have shown us is that in terms of scanner type, field strength, even the scanner OS platform, you need to, if you're picking one scanner, stick to the scanner. And I know that since most of you are going to be working at the NIH, you will pick a scanner and a coil, you'll stick to it. But I think this is relevant to people who might have to look at data from data banks. Like if you're picking it from the Human Connectome Project or the Biobank, they're now acquiring data from different scanners using different coils. And it's just to give you a sense that just because, you can't pull them all together. You need to be mindful of these different factors, right? So that's the scanner-related stuff, and it applies to all sequences. In terms of sequence-specific factors in T1 weighted measures, one constant thing that pops up is the use of things like acceleration. So, so this here is a T1 weighted image. You can acquire it either with the classic MP range sequence or the multi-echo MP range. And if you're using a high field magnet like the 70, you'll be using an MP2 range. Bottom line is you get a really nice white matter, gray matter contrast image, which can then be used for looking at cortical thickness and so on. So what happens with this sequence is that you have the option of using acceleration or not using acceleration. Acceleration is something that you use when you have a very complex coil, like a 32-channel coil. So you have a multi-array coil, and so in the interest of reducing the scan time, you can use different factors of acceleration, and you can get these images at half the time. So what I'm showing you here is without acceleration, and asset here is the acceleration factor for GE scanners. So this is without acceleration, and this is with acceleration. You can see that there are some contrast differences. But what matters is that if you look closer, you can see that this red line, compare this red line with this red line, you can see that the cortical thickness as measured in a non-asset image tends to be thicker as opposed to the one over here. It tends to be thinner. So if you are, so it's also a problem that there are some regions that are more affected by using an acceleration factor versus other regions, right? So these are things that you need to I think, be aware of. Now, on the flip side, if you don't use acid, you now have a 10-minute scan, and if you have kids, you're studying kids, then you're in a lot of trouble because kids simply can't stay still for 10 minutes. So in the interest of getting reasonably good data, what I would recommend is use 2 sets of accelerated T1 weighted images, because just one is not enough. But if you get 2, you can merge them, you can average them, you get better signal to noise, and you can get a better measure of cortical thickness and so on. So that's one of the main issues with T1 weighted images. With diffusion, there are a host of other things that we need to keep in mind. One is eddy current distortions. Eddy currents are those currents that are caused by rapidly switching diffusion sensitizing gradients. And what you see is this, this sort of rimming effect, which distorts the image, and it also alters the actual diffusion sensitization that is being applied. But on the good side, you can correct for this. So Tortoise and even the latest version of FSL offers a very robust solution for correcting these kind of eddy current distortions. The other distortion is the Gibbs ringing. So this refers to these sort of ringing effects that are caused by really sharp image transitions. Well, what this means is in T2 weighted images, such as in the case of diffusion images, you get this very sharp contrast between CSF and the gray matter, and in voxels in the border where there's a sharp transition from CSF to, for instance, gray matter, you get this rimming effect. And this again distorts the diffusivity measures, but fortunately we have a solution for it. If you use the latest version of Tortoise, I believe this corrects for Gibbs ringing. The other sequence-specific factor is EPI distortions, or ecoplanar imaging distortions. So what I have here is a T2 weighted image. This is the same brain, 2 different slices. This is much more inferior than this slice. Now here's what the diffusion weighted slices look like. So if you have the phase encode direction in the anterior-posterior direction, you have the stretching that you see over here, and there's also the signal pileup that you see in these regions. Same, if you do it if you have the phase encode direction in the AP, anterior to posterior, you have a compression here, so it's going in the posterior direction, and you have signal pileup here. And this is if you have right to left and left to right. Bottom line is you get these really very sharp distortions in the image as well as signal pileups. And so this distorts the image profoundly, and one of the things it does is it messes up tractography very badly. Not so much the DTI measures. But again, this is also correctable. So the impact of EPI distortion on tractography was demonstrated by Okan Irfanoglu, where he actually did acquire diffusion weighted images in the anterior posterior phase encode direction as well as the right left phase encode direction, and looked at the trajectory of the corticospinal tract. And this is the uncorrected case. You can see that the corticospinal tract is going all over the place over here. But once you correct it, you get a much more known trajectory of the corticospinal tract. So the key message here is that it's a distortion that is going to be there in most EPI images, particularly diffusion weighted images. And so if you want to do tractography and things like that, you should consider acquiring blip up. One set of images in the AP direction and the other set of images in the PA direction. And you can use either Tortoise or some of the packages like FSL also offers it. But the best, I think so far, the best solution that I've seen comes out of using Tortoise. So those are all the sequence-specific factors. Now, so the scanner-related issues, sequence-related issues, and now let's think about some of the subject-specific factors that we need to think about. So motion, right? So if you think about just about all MRI images are affected by motion artifacts. T1 weighted images are also very badly affected. And the impact of it is very evident in this image. This is a work by Martin Reuter from Bruce Fischel's lab. And what he showed was like measures of gray matter volume as well as cortical thickness are correlated with the degree of motion that subjects have when they're in the scanner. Interestingly, when he also did the calculations to show that 2 millimeters per minute motion results in roughly 1.4 to 2% loss in gray matter volume. Now, it's important to note that there is no real loss of gray matter volume. This is the apparent loss of gray matter volume. It's an estimate that your FSL or any of these packages produce. We think of this as it manifests as a loss because of the distortion, right? And the solution to this is to use, you should talk to, if you're planning a study, the best thing to do is to talk to people like Vinay or other MR physicists who know what are the latest tools, right? So this PROMO stands for prosthetic motion correction. And so if you use the PROMO sequence, you have a better shot of reducing the motion-related artifacts. Also, better instructions, reminding subjects not to move and so on are really useful to bring that down. Motion also, predictably, also has a profound impact on diffusion-weighted images. Here's an image, directionally encoded color map before correction, and this is what it looks like after correction. So it is correctable, but if you can avoid it, you're so much more better to avoid by using, whatever sequence-specific things that you can do. The other subject-specific impact factors are, for instance, blood pressure. So this is a recent paper that I saw where they compared T1 weighted images. They did voxel-based morphometry on different groups of people with different levels of hypertension, and they showed that people with hypertension tend to show a reduction in gray matter volume in these regions. Now, and obviously this has an impact on cortical thickness also. So if you, this might not have anything to do with sort of an atrophy related to hypertension. It's just that the blood flow and blood flow related artifacts can change the MR signal, and which might manifest as a decrease in gray matter volume or a decrease in cortical thickness. So that's blood pressure on T1 weighted images. With diffusion weighted images also, you have issues related to the cardiac cycle. So here is the directional encoded color map generated based on looking at a single subject's images that were acquired during the diastole phase of the cardiac cycle. And here's the same subject, but now these images are acquired from the systolic phase of the cardiac cycle. Notice that one of the biggest tracts in the brain, the corpus callosum, sort of disappears if you just look at the systolic phase. The image is constructed from the systolic phase. So just to show that there are some regions that are very badly affected by diffusion, by the cardiac pulsation artifact. This here is another map. Basically, it's a fractional anisotropy map overlaid with the red areas indicate regions that are at risk for being spurious, because of the cardiac pulsation artifact. The solution again here is to identify these outlier voxels. There are very principled ways of looking at picking up these outlier voxels and replacing it or removing it using packages like Tortoise. So those are all the subject-related factors. The other issue, so now you have, let's say you picked a scanner, you've acquired some data, everything went well, you now have data. You're going to analyze your data now. First off, it's useful to remember that things like free server version and even Linux version, these kind of things also matter. So it turns out this paper suggested that you're like using the same data, the cortical thickness measures that are being reported based on a Linux machine versus a Mac OS machine, there were subtle differences. Now these are really subtle. I mean, but it's still something, it is a nuisance variable that you want to control for. So if you are going to start on a project, stick to the same operating system and same version and so on. Now, so you install the software and let's say you're running a voxel-based morphometry analysis. The important thing to keep in mind here is that from this state where you have raw data to this state where you have like statistical maps computed, there's a lot of stuff that's going on. The data undergoes several transformations. So the data gets normalized to some standardized template. There's gray matter segmentation, then there's modulation of gray matter, and then you smooth it with some sort of a smoothing factor, and then you get these results. You do the stats there. So biases can be introduced at any point in this pipeline, and that can have profound consequences. So this was demonstrated by Derek Jones, who looked at the effect of filter size. This is the Gaussian filter that you use for smoothing the data, and how it changes the results, and in turn has an impact on how you make inferences about results. So that's things that you need to keep in mind. Another thing to bear in mind is with the same data but with a different software, you can produce different results. And this is work done by Adam Thomas here. And so Adam looked at a study where he was interested in looking at how, with learning a new task, what are the behavioral changes, what are the functional changes, and what are the structural changes. So he found evidence for behavioral changes as well as functional changes in the brain. When he looked at the nature of the structural changes following training, with SPM2, he found results like these, but with everything being the same but a different package, he found entirely different results. Now these are very primitive versions of the software. Many of the bugs have been corrected, but what I've learned from working with software engineers is that the software is an organic being. It continues to evolve, so there are bugs being fixed. You need to be mindful of these kind of things, and I think it'll be very useful, or it can completely throw you off. So the solution here is to be aware of these issues, test the reproducibility with different packages, and also talk to your image processing guru or your statistics guru to figure out what are the right kind of statistics you need to do, given all these issues that are in play. With one of the important parts of doing like a whole brain voxel-based analysis is to do spatial normalization, where you bring all your subjects' data to a standardized space. Now there are several ways of doing it. The traditional way of doing it in diffusion images was mostly to take the fractional anisotropy map and then run it through the standard affine nonlinear registration techniques, and then you have everything in the same space, and then you do voxel-wise statistics. Now, there's another way of doing the same thing, but which is much more efficient and probably much more robust, and that's to use the full diffusion tensor. So what I mean here is that if you have, let's say, a single subject, these are two subjects, A and B, and you have 5 sessions of data, you take, and these are in the native space, right? So you take their data and you make a template out of this. You can make a template out of all this single subject's data. So you make 2 templates, and then From these templates, you make a template of the templates, right? And so you have transformations going from this template to these individual images, as well as from this super template or the study template and all the way back to this. Now you can apply these two transformations to the data in the native space, bringing all of them into a standard subject space. And you're doing it at the diffusion tensor. And so once you have everything in the tensor form, you can then extract these FA and radial diffusivity and all these other maps. And that I think it's a much more robust way of bringing your data into a standardized space so you can do voxel-wise statistics. So to summarize, some practical recommendations: pick a stable scanner and stick to it. Pick a robust MRI sequence, talk to an MRI physicist about your study goals, what kind of people you're going to study, how much time you have, and so on. Better data comes at a cost, so if you want motion-free T1 weighted images, you'll have to invest at least 10 minutes. So if you cut corners, you will have to live with those bad data. You want to pick a stable pipeline for data processing. There are many that are around. FreeSurfer is now probably the most stable software for surface-based analysis. For voxel-based morphometry style of analysis, you have FSL and SPM. Many of the issues have been sorted out with these packages. For diffusion pre-processing, I would highly recommend using Tortoise because it gives you the ability to correct for a whole host of distortions. This was put together by the people who invented the diffusion tensor, so they really know what's going on and how to deal with these issues. So I have learned a lot from working with this group of people. I would also recommend QCing images as you collect them at the scanner. Now, so you can see these images popping up in real-time reconstruction. It's useful to keep an eye on it. So if you find some sort of artifact, you want to make sure, sneak in another 5-minute scan, get the other data, because once the subject leaves, all bets are off, right? So might as well do it while you're there. Once you process the image, it's worthwhile to QC it after. And be consistent with your instructions to the subject. If you tell one subject, oh, you go to sleep, and the other one, stay awake, or if you don't do any of these things, you might as well, there might be nuisance-related factors, like more motion if someone's sleeping, like they tend to wake up suddenly and that kind of stuff. So you want to be consistent with your instructions to the subject. Here are some really nice papers that I think give you a very broad idea of all the other methods that are out there in terms of using it to look at brain changes, neuroplasticity, and so on, and the sort of issues that you need to be mindful of. Right, so we've looked at the popular MRI methods. We now know the limits of the challenges that we have, and we also know how to overcome some of these limits. So what's the most important tool for imaging brain changes? So what I think is most crucial is really a simple thing: good experiment design, right? And here's why I think this is very crucial. So when you start thinking about what are the candidate brain structures that are likely to change when you're trying to use some sort of an experimental paradigm, it can be a whole host of things, right? It could be neurons, dendrites, dendritic spines, synapses, axons, axonal boutons, the glial cells, capillaries. A whole bunch of things are likely to change. And especially if you're trying to pick up something really subtle, you're not only trying to image the structure, you're trying to image a change in these structures, which are already tiny, right? So the challenge is MRI robust enough to detect subtle structural changes, given what we know of all these limitations, right? This is sort of the question that I was sort of asking a while ago. And one thing that I learned from looking at animal models is that they have the luxury of very super high resolution, but they don't have the ability to look at the whole brain. So for instance, here's a really nice study that I like. This is Zhu et al., where they trained these mice, adult mice, to retrieve a pasta using one particular arm. So it was lateralized training. And the key finding was that compared to a control group of mice, the training mice show an increased proliferation in dendritic spines. So they concluded, compared to the control, from day 0 to day 1, day 0 to day 1, you see an increased proliferation of these new spines on those dendrites. So they concluded that motor learning in the adult brain is mediated by changes in dendritic spines. So this is with 2-photon microscopy. You can image things at a single micron level. You can do this, right? But what I really liked about it was the design, right? So here they were able to show that this change was specific to the trained group, it is specific to the task and the brain region. So here's a plot where the training group, which is in black, and relative to it, the control. So this is the number of days in the imaging interval. So 2 days after training, 4 days, 8 days, 16 days. Right, relative to the control, you can see a big spike in the fast spine formation. Here, they're comparing different other animals. So a group of control mice, a group of mice that failed to learn, a group of mice, or the same mice, but in the opposite hemisphere and so on. So by doing these different comparisons, they make a strong argument that this is specific to the trained group, the task, and the brain region. And they're doing the comparisons. So human MRI has lowest spatial resolution, and it has poor biological specificity. When you image the brain, you're not imaging dendrites or spines or any of these things. You're getting at a mesoscale level the characteristics of certain brain tissue. So the questions that we can ask then are, is there an experiment-specific change? Where in the brain are the changes? And you can also ask, what is changing? It is a little difficult to address, but if you use like different modalities, MRI modalities, and bring them all together, we have a better shot at asking these kind of what is changing questions. It's not impossible to do. It's difficult. It requires a lot of investment of time. So these are things that we reviewed in a review paper, and we thought of coming up with a framework for sort of assessing how robust training-dependent structural changes are. And so one way to ask is, even if you're designing an experiment or if you're reading a paper that describes results about how training changed the brain, you can ask if the specific changes, if these changes, are they specific to the training group, are they specific to the task, or are they specific to the brain region, right? So we looked at a whole bunch of the existing MRI studies, and the classic design is a longitudinal design, partly because if you have a longitudinal design, you have a pre and a post, and you can say you can draw a causal connection between what the experiment did, right? So you have a pre-training scan or a baseline scan, and then you give them some training, presumably some structure changes, and then you scan them after. And then you do voxel-wise statistics, and before versus after, you have some difference in the MRI measure. Can we then conclude that training has changed the brain? The problem here is that is measurement error, right? So this entire thing could be due to some confound or some scanner measurement error and so on and so forth. So what you really need here is a control, right? If you look at some of the literature that's out there, people tend to think that we're imaging structure, and so we don't need a control. This is, it's like you're taking pictures of how much can it change. So, but at the core of it, if you keep in mind that this is MR signals, it's not structure, it's an MR signal, and MR signals are bound to change because of all these other factors that I talked about, then you really want to have a very good control, right? So the alternative is to have a control group, and that helps you to look at a task like a group by time point interaction, and you can do voxel-wise statistics, and you might be able to find something more convincing. The other thing to do is to, just like in the animal world, so here's a case where Van Praag et al. Were looking at neurogenesis. And the claim that they wanted to make was that running evokes the proliferation of new neurons in the dentate gyres of the hippocampus. Now, it's specific to running, and the reason they can do this is because they compare it relative to a whole bunch of different tasks, right? So enriched environment, swimming, learner, and a standard control, right? So when you have different controls, you can actually make the case that increase in neurogenesis is specific to running. Now, here's a human imaging case where the conclusion here is that 11 hours of IBMT is integrative body-mind therapy. 11 hours of IBMT increases fiber integrity in the left anterior coronal radiata. And this is relative to a group of control. So they have a control group, but the problem is that if you don't give the control group a task, there might be confounds that are emerging from that. So a clear confound that I think hasn't been taken care of here is that one thing that changes with IBMT or any sort of mind therapy is breathing or blood pressure, right? So if they're controlling, if the benefit of IBMT is that people are controlling now their breathing exercise, they're using breathing exercises to control their breathing, they're going to breathe like that in the scanner. And we know that things like cardiac pulsation and respiration also have an impact on the data. So a good control would be something like yoga or something. So you have two groups, one which is yoga, and you're making and that allows you to make the claim that IBMT has a much more stronger influence on brain structure than another kind of training exercise. And the other final issue is, as I said, about the specificity to a brain region. If the effect of training is specific to a brain region, in order for you to say that, we need to do the actual test. So some of the studies that we reviewed would point out that, look, the pattern, so this is a measure of gray matter density at 3 different time points from this region. This is from the left hippocampus. The left hippocampus shows a linear change as they studied for some exam, while this sort of increased initially and then plateaued. Now, if you want to make the contrast between these two brain regions, then we need to actually do the statistical test. So it's important to actually run a group by time point by region kind of interaction analysis and see if that holds true. So we took all of this sort of principles that we had in mind and decided to put our money where our mouth is. And we decided to do this experiment where it was a longitudinal design. And the question was, how does learning versus mastering a motor skill change the human brain? So here, a group of 20 people, what we did was we scanned them across 4 different time points. The first visit, we scanned them at 10 in the morning, give them 2 hours of rest, and then scan them again for 2 hours. And this was just rest, or what we call as looking at measurement error, intrinsic measurement error. A week later, we got the same subject back, scanned them at 10 in the morning, then gave them a right lateralized training video game, and then scanned them again. 1 week later, we scanned them at 10 in the morning. We gave them now a left lateralized motor sequence training task. And then after 2 hours, we scanned them in the PM. So this allows you to look at acute changes due to training. So also this training task serves as a control, an active control for this training task and vice versa. But you also have a rest control built into the design. A week later, we scanned them again at 10 in the morning and 2 in the afternoon. But this time we gave them between visit 3 and visit 5, during the weekdays, we gave them 1 hour of training for the 5 days. So this allowed us to compare long-term training and short-term training. And during each one of these training sessions, we acquired multimodal MRI, T1 weighted images, advanced diffusion weighted images, resting state fMRI. We also got like quantitative T1, T2 measures. We got 2 data sets of each, so we can test for reproducibility of these findings and so on. When we looked at the total brain volume, what we saw was this is taking the T1 weighted images, we're running it through the FreeSurfer pipeline. And one of the postbacks looked at how how stable is total brain volume. And what he found was that there's a fluctuation in the total brain volume from AM to PM. So in each of the visits, AM to PM, you have the sawtooth pattern. And then given that this was most obvious in visit 1 and visit 4, when we didn't have any training, we decided to look just at visit 1 and visit 4. And what we found is that roughly 16 out of the 19 participants show a reduction in total brain parenchymal volume. So we call this the time of day effect. Now, this is nothing new, because there are other studies that have reported this. with a very large sample size. But what we found is that even in a small group, when you have like a very systematic design, this tends to show up quite profoundly. Now, you could make the argument that in one sense, we made a classic experiment mistake, not randomizing the time of day. But we were stuck with a certain kind of time slot, and so that's something else that you need to think about. But in any case, this was a systematic thing that we found. Then we started digging into it further, and we looked at, so this again shows the same subjects at 2 different time points from all the 4 scans. And you can see there's a significant reduction in volume that's normalized to total intracranial volume. That's roughly 2.29% of the intracranial volume. Turns out that the bulk of it can be explained by a reduction in total gray matter volume, as well as a subtle reduction in total white matter volume. But while these 2 tissue classes, you see a reduction, with CSF, you see an increase. Then we looked at the second-order measures like cortical thickness. And what we found is that, indeed, this also has a really profound impact on measures of cortical thickness. And these are the regions where cortical thickness tends to decrease significantly from AM to PM. Obviously, given the, that we use the same images for doing voxel-based morphometry, we also see a significant reduction in gray matter volume in these same regions. Now, it's not specific to just T1 weighted images. When we looked at the diffusion weighted images that we acquired from these same study, we found that while from AM to AM, so these are comparisons of scans from that are 3 weeks apart, you see, like, if anything, specks of different reduction in mean diffusivity or trace. These scans are just 2-3 hours apart. And you can see there's a big reduction, an increase in mean diffusivity from AM to PM. And this is also in the same regions where we see a reduction in cortical thickness and gray matter volume and so on. And this here is just to show you that the benefits of having blip up and blip down. So these function as 2 independent data sets, and you can test for the reproducibility of your findings, and so on. So I haven't shown you anything about the training because that's still a work in progress. What I wanted to point out was that the rest controls in this case helped identify time of day as a significant confound. So this, I think it's really important to keep in mind for longitudinal and cross-sectional designs. And even in cross-sectional designs, we might be in the circumstances where your patients, because they are probably more available during AM in the day, and because the healthy controls tend to come to scanner after their work, you might have a systematic bias in how you scan your 2 groups of patients, and you might have differences purely because of that, rather than any pathology. So even cross-sectional designs, I think it's an important thing to keep in mind. Time of day impacts, T1 weighted measures, DTI measures, and I haven't shown you this, but even resting state measures are affected. But by having these different measures, we sort of get some insight into what the underlying mechanism is. So DTI, we used a slightly advanced diffusion imaging. So this allowed us to look at to what extent CSF or some free water is contributing to this effect. And we were able to show that this is indeed something related to CSF. With the resting state, we are finding large changes in functional connectivity. So there's some suggestion that there's some blood flow related changes also that are associated with the time of day effect. So having replicates of data also helps us to test reproducibility. And so from having learned from this experience, we are now looking at it in terms of modeling it as a time of day by visit interaction, rather than looking at it as a visit by visit type analysis. So to summarize, not only do you need a control group, you need a well-matched control group with the control group having an effort-matched task if possible. It's always useful to use multimodal MRI data, collect 2 sets of data if possible, and consider all these potential confounds. So there are different biological rhythms that one needs to think about, like the circadian rhythm. The post-lunch dip, it's a real thing. People are much more slower when you scan them immediately after lunch. Even behavior tends to slump a lot immediately after a big meal. There are other cycles, the infradian cycles, and other seasonal cycles. So there's a recent paper which showed that the hippocampal volume changes based on the seasons. And it might be related to some sort of primitive mechanism, but there are a lot of things that one needs to be mindful of. The other thing is levels of hydration. Are people drinking too much water? So if you have a subject who comes to your scanner biking or running, he's going to come and drink a lot of water right before your scan. He's going to mess up your data, or things like that. You want to understand where these subjects are coming from. Are they having? So if you have morning scans, you have a risk of highly caffeinated subjects, and so on. So what I'm saying is there's so many of these issues, and if you can't screen or control for it, measure it and be mindful of it. That's what we can do. And report the methods you use it in as much detail as possible, so someone else can replicate it. The other thing is that it occurs to me that these second order measures like cortical thickness or gray matter density and these kind of terminologies, we need to bear in mind that these are just estimates that are derived from the MR signal. And I've mentioned this again, but I'm going to restate it just because what we're measuring is the MR signal. We are assuming that this is telling us something about cortical thickness or white matter microstructure. But anything that changes the MR signal will change your cortical thickness and so on. So that's one other thing that you want to keep in mind. These are two papers that have been very useful, that I find very useful to understand how to approach imaging brain changes. And finally, thank you for your attention. And there are many of the slides are from people that I've worked with. Amrita, Adam have taken some slides from your papers and so on. So thanks to all the members of these two labs. Thanks.